Familial hypercholesterolemia is the most common form of heart disease. It is associated with increased LDL levels in the bloodstream, also known as bad cholesterol. About 1 in every 250 Americans is diagnosed with FH each year, leading to a premature risk of heart attack and stroke. But what causes FH? Every person has a unique sequence of genetic code that plays a role in their physiology. With the increasing use of genetic sequencing as a diagnostic tool, the ability for doctors to bridge the gap between genotype and phenotype becomes smaller and smaller. But one problem arises. How do doctors interpret the consequences of their genetic code? Let's take a closer look at some hepatocytes in the liver. The protein mainly responsible for lowering LDL levels is the LDL receptor. It binds to LDL, gets engulfed through endocytosis, degrades the bound LDL, and is then recycled back to the cell surface. But what can lead to increased LDL levels? A protein known as PCSK9 binds to the LDL receptor. Upon endocytosis, PCSK9 traffics LDLR to degradation, indirectly increasing blood LDL levels. But how can we understand variations in PCSK9? Let's mutate. Say we change amino acid position aspartic acid 374 to tyrosine. What is the effect? This PCSK9 variant appears to have increased binding efficiency to LDLR. A person with this variation could be at risk for development of FH. With a complete variant effect map for PCSK9, we will be able to provide evidence strengths towards benignity and pathogenicity, enabling doctors to better treat patients.